In today's lecture, we're going to continue talking about somatosensation, picking up on where we left off when we were discussing touch and proprioception, but today we're going to be discussing pain and temperature sensation, and how does the body sense pain, how does it sense temperature, and what are the pathways involved that allow us to perceive these things. So, to continue on the theme of this first part of the course, where we're discussing the sensory pathways in the brain, every sensory pathway is going to have some specialized receptor cells, and since we're talking about somatosensation, that involves some specialized nerve endings. So we will be discussing a little bit more of that today. They're not nearly as interesting or complex as they are for touch, but they do have some really interesting things happening chemically. So why don't we get to it? So as we've been talking about, somatosensation is conveyed primarily through receptors located in the skin. And in the last lecture, we discussed all the different kinds of mechanoreceptors, and these are used to sense changes in pressure or movement. Today, we're going to be talking about the nociceptors, which respond to pain stimuli. They sense changes in pressure, but acute pressure. And we will also be talking about thermoreceptors, which respond to changes in temperature. So these will be the two that we'll be discussing today. So nociceptors, they don't have any specialized endings like the mechanoreceptors. Instead, they are just free nerve endings that sense when tissue is damaged or are at risk for damage. They tend to be sensitive to shearing, changes in pressure, so they do have some stretch capability to them, but there are a number of other things that can activate as well. So it's pain, we can define as the perception of uncomfortable sensation. That can include irritation, soreness, stinging, aching, throbbing, even itch, following nociceptor activation. So what kind of stimuli can cause tissue damage? There's all kinds. So when you get a tissue injury, like a splinter, it causes me pain just to look at that. There is a mechanical stimulation that happens, and that can induce change in these free nerve endings. So there's the stretching that occurs similar to the free nerve endings for touch. But temperature extremes can also have an influence over these cells. So we'll discuss the channels for that as well. Oxygen deprivation can in fact induce pain sensation. Exposure to certain noxious chemicals will induce pain sensation as well. And substances that are released by damaged cells. So if a nearby cell is damaged, it will undergo a process of lysing, and material will spill out of it. It'll activate certain chemicals if it's irritated. Macrophages can release certain factors, so the immune system will cause that irritation in the skin, some swelling, and that will act on then these receptors, the free nerve endings, to induce activity to cause them to fire. So there are a bunch of different factors that can lead to firing of these free nerve endings. So there are a variety of stimuli that can trigger these painful sensations, and not surprisingly, there are a variety of receptor types that process pain-related signals. We're going to talk a little bit about a few of them. So this is an example. This is a picture of a cartoon of a receptor called TRPV1. The TRPV1 stands for thermotransient receptor potential vanilloid receptor 1. Better to just say TRPV1. It can be activated by several different stimuli. So it is heat sensitive, so this is a temperature sensitive channel. It is sensitive to high concentration of hydrogen ions, so sort of like the sour tasting cells in your mouth. these will detect, like, if you spill a bunch of acid on your skin, and so it'll detect that as well. And then it also will pick up capsaicin. So capsaicin is a ligand that has specific sites onto these receptors, and it'll induce them to open and allows sodium, calcium to flow into cells to depolarize the cell membrane and induce action potentials. So this is a ligand-gated ion channel that permits the entry of sodium and calcium ions. These are example of different chilies that can induce activation of these receptors. There's some recent research, just to refer to this, showing that these receptors are also sensitive to physical deformation, so they can even pick up pain via stretching. One of the things that's interesting about these kinds of receptors is that so the TRPV1 receptor comes from a family of receptors that are temperature and chemical sensitive. So when we have capsaicin around from peppers, the TRPV1 receptor is activated. We also can activate this receptor via exposure to heat. And here, this is showing the temperature range that can induce activation of this channel. So, interestingly, the reason why we refer to spicy foods, like things from peppers, wasabi, those kinds of things, especially from peppers and those with capsaicins in them, we refer to those as feeling hot. Oh wow, that food is like super hot. I can't possibly eat it. It's way too hot for me. We're using the word hot over and over and over again when the food could be at room temperature or even cool, but it will taste hot because of the capsaicin, and it'll be activating these channels that will induce a feeling of hot, because the same stimulus, these same channels can be activated via capsaicin or temperature, and we can't really disambiguate the two. We can tell a little bit because of the mix of channels, but that pain will be induced via administration of chemical or heat. And so one thing to keep in mind is if you are going to be eating like some crazy hot spicy thing, there's something called the one chip challenge, where it's like Carolina Ghost Reaper pepper, I believe, and in this package there's a single chip with a whole bunch of this pepper on it, and the one chip challenge is you open up the package and you're supposed to eat the chip. That's it, without passing out or throwing up or whatever. And there's videos on YouTube that are super hilarious of people eating these things, but there's one in particular where they were doing this on a morning news channel, and they handed these chips out to all the people, and one of the anchors, so this is the morning news, right? It's a local morning news thing, and they're going to have coffee near them, right? Because it's like 6 a.m. in the morning, and they're doing this crazy one chip challenge, and they were not prepared for it. So this one news anchor, she takes the chip, and it's just way too hot for her, and she's trying to wash it down, so she takes her coffee and tries to wash it down with coffee. You could imagine the problem there is now she's swamping not just these TRPV1 receptors with capsaicin, but also now hitting them with heat, and it just makes it that much worse, and activating additional thermoreceptors, so it's just more and more and more hot, and she starts heaving and throwing up, and it's pretty gross and sort of hilarious at the same time. Anyway, don't mess with your TRPV1 channels if you can avoid it. So these channels, they come in a bunch of different flavors. These are just 3 of them. There's I think upwards of 10 or maybe even more of these types of channels that have been identified in vertebrates. There are several of these heat and cold sensing channels in invertebrates as well. And in fact, Lena Nee's lab, who is part of our department, this is what she studies. So she studies temperature sensation in Drosophila, the fruit flies, and she's working on characterizing these heat sensing channels, how fruit flies can sense heat. So very interesting stuff, and I encourage you to look at her research if you want to learn more about these. But we're going to just talk just a little bit more about them. So I mentioned the TRPV1. There's also TRPA1, which is sensitive to active ingredients in mustard and wasabi. This is also a painful stimulus, but interestingly, TRPA1 is activated by very cold stimuli. So unlike TRPV1, which is activated by heat, this is activated by cold. So, but wasabi and mustard are painful stimuli. So this isn't really telling us that it's cold. This is just telling us that it's noxiously cold. That this is so cold that it's painful. We do have other receptors that actually mediate the feeling and sensation of cold, like the nice cold, which would be TRPM8. So this is a channel that is sensitive to relatively cool temperatures. So starting below like around 28 degrees and going down and sort of turning off by about 10 degrees, which is different than TRPA1, which is sensitive to much colder temperatures. This channel is pleasantly cold, and this is sensitive to menthol, which is the active ingredient in mint, winter mint. So if you look at any kind of advertising with things that are mint flavored, it's almost always that they have cold and wintry scenes, and it's because the channel that is activated by menthol gives us a sensation of cold because it is acting on cold receptors. So it's really amazing like how flavors have this masking and informative content that is tied in with temperature, and how our connection to temperature helps shape our perception of the foods that these ingredients are found in. Really very cool stuff. So capsaicin, in fact, can be used as a tool for inducing pain. Since it activates these specific receptors, you can induce pain. This is a figure taken from a paper published just a couple of years ago, looking at the activation of TRPV1 channels and its relationship to the progesterone receptors. So this paper goes into a lot of the molecular mechanisms, but this is a figure from one panel from their first figure where they're trying to describe the phenomenon. So there's this idea that when an animal or human is pregnant, that they have lower pain sensitivity, that they can tolerate greater amounts of pain. And so we have a couple of different categories here of this treatment. We've got like two different dimensions in which these animals are being treated. So these are mice, and they have, in green, these are non-pregnant mice, okay? And then in red, we have pregnant mice. So that's one dimension of the categories, non-pregnant and pregnant. Then the other dimension is we have animals that were injected with just a vehicle. So this would be the solution that capsaicin would have been diluted into, but there's no capsaicin, hence this is a vehicle. So this is a negative control. And an injection itself is going to cause some discomfort and some pain, but it's not that much. And what you can see is that some of the animals, they spend some time licking their paw because, like no one likes to get injected. But if it's not a noxious thing that's being injected, it's really not that big a deal. And you can see there's really no difference between the non-pregnant and the pregnant mice here. But when you use capsaicin and you're inducing the TRPV1 channels to be activated, so they're injecting capsaicin into the paw, you can see that both groups are now licking their paws more than if they weren't injected with capsaicin at all. But interestingly, the pregnant mice have a lower amount of time licking their paw relative to the non-pregnant mice. It's nearly half. So this looks like the pregnancy is conferring some sort of pain tolerance that is not present when mice are not pregnant. So there does appear to be some sort of advantage to being pregnant and having greater pain tolerance. So I'm showing this and I'm going through this, not for you to take this as something that's really critical for you to learn. I just think it's kind of an interesting example of something that you might find in a paper that is showing capsaicin being used as a tool in order to induce pain that will last a little while and then disappear. And it's a pain that can be delivered in very precise amounts. The other reason why I'm showing this is because I wanted to provide a little bit more detail on the video project. So this is like the one big project that we'll have in this class, and you will be expected to make a video like the one that you are watching, presenting one research article. So you have to just find a single research article. I'm giving you plenty of advance notice for this. It is due Thursday, December 3rd, the week before the week that finals will start. And what article do you get? What article are you going to present? Well, you get to decide. You can pick any paper that is from one of these three journals: the Journal of Neuroscience, Nature Neuroscience, or Neuron. And it needs to be published within the last 3 years. And as I've said before, all three of these journals, they publish every single week. And there's usually 15, 20 articles in each issue. So between the three of them, you have more than 100 articles published every single month. And you have 3 years to choose from. So there's plenty to choose from. We'll talk a little bit more about searching for articles in just a moment. But the format of the project is this. You have a 10-minute video, and the emphasis should be on the introduction, the results, and the discussion. You do need to cover methods, but you don't need to go into crazy details on the methods. I don't want you to spend 8 minutes talking about what they did. I'd rather you focus on giving a good introduction, then discussing a little bit about what they did, and then maybe some results. Then you might talk a little bit more about some methods and some additional results. You might go back and forth. We'll talk about more of that later. And then finish with like a minute or two of discussion. And this is going to be an individual project, not a group project. Where can you find primary research articles? This is the way that scientists communicate their latest and greatest research to the public and to each other. One excellent resource is called PubMed. This is a database maintained by the National Institutes of Health. Your tax dollars pay for this resource. It contains over 30 million citations, 30 million different articles across biomedicine, then from thousands of journals going back decades. It's an excellent resource. You can type in any topic that you're interested in into the search term here, and it will pop up, and you will have hundreds of articles to look from, unless you get really specific. But it'll include all the journals, including the really crappy ones. So in this course, I'm wanting you to understand and appreciate that there's different levels of science journals. The top neuroscience journals are the Journal of Neuroscience, Nature Neuroscience, and Neuron. There are others, but these are probably, most people would say that these are like the top 3 journals in neuroscience, specifically for neuroscience. For this assignment, you can go to any of these websites, one or any of them, and just type in search terms if you'd like. If you're interested in, say, alcohol, you can type alcohol in there and you'll get the latest and greatest research related to alcohol. Schizophrenia, Alzheimer's, depression, TRACT1 receptors, the coronavirus. You can get the latest information from these websites. Now, the other nice thing about these websites is that each of them have a pretty good editorial staff, where they also will include opinion pieces, background information on some of the most exciting articles that's being published there. And they publish these every single week. So you can go week to week and just see what is being published and what kind of exciting things are happening in neuroscience. Journal of Neuroscience, they have one
um, feature that they do, which is called Journal Club. And this is a place where graduate students and even undergraduates can review an article, either published in the Journal of Neuroscience or published in some other journal, say in Neuron, that these students are trying to highlight as being interesting and cutting edge. And so these are nice, simple summaries of interesting articles that have been published. That actually might be a good resource to look at if you are sort of a little worried about looking at some of these research articles. That would be one place to look.
All right, so back to the topic at hand. Um, that's we've already covered what I want you to know as far as receptors in the skin for pain. The receptors are not super special, at least morphologically. There's not a bunch of different types, but there are several different categories of receptors that do mediate certain kinds of pain, such as chemical pain or temperature, and there tends to be cross modality across them. We're also still learning a lot about pain and pain sensitivity in the skin. There's still a lot that we don't quite know, but we do know how it gets up into the spinal cord. So, and I wanted to spend a little bit more time reviewing the spinal cord again and talking a little bit about the anatomy of the spinal cord. So the spinal cord, it carries this information between the brain and the body. That's its job. It transmits signals to and from the skin, from joints, from muscles, from the viscera. The parts that we're talking about right now are the dorsal roots. Those are shown in red. So this is on the back side of the spinal cord. We will talk about the ventral roots when we talk about motor output. But the ventral roots, here in red, they have these dorsal root ganglia, again, emphasizing the ganglia here, and that's where the cell bodies are located. And that these sensory fibers, they will either go up the spinal cord, or the one that we're showing here is that these fibers will synapse onto neurons in the dorsal horn. So within the spinal cord, we have fiber tracts on the outside. That's what this lighter part is. So these are all axons going up and down the spinal cord. And then we have a bunch of cell bodies in the middle. It kind of looks like a butterfly here. They're actually, they're referred to as horns, so I suppose you should probably think of them as horns. And there are dorsal horns, this part going up into the dorsal side, and there are ventral horns going down to the ventral side. So the pain input comes in through the dorsal side and synapses onto the dorsal horn. So let's talk a little bit about that. All right, so back to the topic at hand. We've already covered what I want you to know as far as receptors in the skin for pain. The receptors are not super special, at least morphologically. There's not a bunch of different types, but there are several different categories of receptors that do mediate certain kinds of pain, such as chemical pain or temperature, and there tends to be cross modality across them. We're also still learning a lot about pain and pain sensitivity in the skin. There's still a lot that we don't quite know, but we do know how it gets up into the spinal cord. So, and I wanted to spend a little bit more time reviewing the spinal cord again and talking a little bit about the anatomy of the spinal cord. So the spinal cord, it carries this information between the brain and the body. That's its job. It transmits signals to and from the skin, from joints, from muscles, from the viscera. The parts that we're talking about right now are the dorsal roots. Those are shown in red. So this is on the back side of the spinal cord. We will talk about the ventral roots when we talk about motor output. But the ventral roots, here in red, they have these dorsal root ganglia, again, emphasizing the ganglia here, and that's where the cell bodies are located. And that these sensory fibers, they will either go up the spinal cord, or the one that we're showing here is that these fibers will synapse onto neurons in the dorsal horn. So within the spinal cord, we have fiber tracts on the outside. That's what this lighter part is. So these are all axons going up and down the spinal cord. And then we have a bunch of cell bodies in the middle. It kind of looks like a butterfly here. They're actually, they're referred to as horns, so I suppose you should probably think of them as horns. And there are dorsal horns, this part going up into the dorsal side, and there are ventral horns going down to the ventral side. So the pain input comes in through the dorsal side and synapses onto the dorsal horn. So let's talk a little bit about that. So here, now we're talking about the blue afferents. Last time we talked about the red ones, which would be the mechanoreceptors. The blue one here is pain and temperature. And this is then, so this, let's say it has TRPV1 in the, out here in the free nerve endings. You have a little cut here, so it's already irritated, and then you start chopping some peppers, and then that pepper juice gets in there, and now it's really, really hurting. And so then you wash your hands with some really hot water, and now you're really causing some crazy pain, because now you got irritation, chemical, and temperature all acting on the same receptors. So these neurons are firing like crazy. And you're getting pain and temperature information carried up. Here's the cell body, and here in the dorsal root ganglion. And then it goes into the dorsal horn. So unlike the mechanosensory fibers that just then turns anterior and goes up towards the brain, this neuron actually synapses on another neuron that sits here in the dorsal horn. Okay, so now we have a different neuron. And then the decussation for pain actually happens at the level of the dermatome that's being innervated here. So in this case, we're looking at the finger. So this is one of the dermatomes, like at the cervical side, the last cervical portion. I think that's C7. And then you have decussation happening here. So there's this neuron then crosses the midline and then goes up a different fiber tract, different than the mechanoreceptor tract, and then goes all the way up to the brain that way. So this is an important point, so I want to go over it just again briefly. So the nociceptors are present there in the skin. There's some in bone and even organs and things. There aren't nociceptors in the brain, though. Interestingly, you should know. So you can cut into the brain and you will have all kinds of problems behaviorally, psychologically, maybe sensory, but you won't be experiencing pain. So here are the cell bodies located in the dorsal root ganglia. And then it goes up on the contralateral side of the spinal cord. We talked about proprioception fibers last time. Those go really, really fast. And then we have the pain fibers. They are different. They have a much smaller axon diameter. Compare, maybe 1 micron diameter to 20 microns diameter. We have two different kinds. We have the alpha delta fibers, which are myelinated, but only like not thoroughly myelinated, like the proprioception and touch fibers. And then we have C fibers, which aren't myelinated at all, and in fact are actually pretty slow. We know that pain comes in two phases because of these two different fibers. Because this one is myelinated, it's actually pretty fast. You've got 5 to 30 meters per second. This is going to get up to the brain pretty quickly. So these fibers, they provide what we call first pain. So when you stub your toe, you have this immediate pain sensation. Those are the alpha delta fibers that are carrying that pain sensation. Then we have second pain, which comes really within a second or two typically, but that's a pretty long time as far as nervous systems go. This is like 20 milliseconds in, maybe 50 milliseconds in. This is like maybe half a second or even a second or even longer. These are carried by the C fibers. And if you knock out, we know that if you knock out the alpha delta fibers, that you only have second pain, or if you knock out the C fibers, you only have first pain. So we know that there are two different pain informations that are carried to the brain. Another important concept with pain is that there's something called referred pain. So the pain fibers from the viscera, like from the heart or the gut, they enter into the spinal cord via the same route as the cuteness receptors, nociceptors, the cuteness referring to skin. So thus, skin and visceral pain input is mixed in the spinal cord. And what this means then is that there's crosstalk between these fibers. And that our body, we have this somatosensory map up in the brain, it doesn't have parts that are specifically designated for the gut. And instead, the pain in, say, the gut or the heart will induce pain in which we perceive to be on the skin. And we refer to this crosstalk as referred pain. So when visceral nociceptor activation is happening, we perceive it as happening on the skin. So for adult males especially, they may feel pain sensation on the left side of their body, and especially in the arm. That can be an indication that you're having a heart attack, and you'll feel that pain there. Urinary bladder pain sensation will be perceived in the urogenital and anal areas. Prostate. So this is a very small gland that's up there in the rectum, and people who are having prostate problems, they can feel this pain all the way down in their foot. It's crazy. So these are areas that we know the body perceives as being on the skin, and physicians will use this information to try to pinpoint maybe what's going on inside the body. So we're going to be talking about pain's pathway through the body, and it's the route that it's going to take. So pain information, we're showing two different routes here through the lower body or the upper body. You can see that these neurons are, the cell body is located in dorsal root ganglia on this individual's left-hand side, I guess, or right. And it's going in through the dorsal horn and then synapsing onto neurons in the dorsal horn. Okay. Right? So afferent fibers, they are synapsing there onto these neurons. So the first thing we have is the nociceptive signal causing an action potential to then synapse onto these neurons. On those second order neurons within the dorsal horn. These second order neurons, like I said, they decussate and then they go up the spinal cord. And the path that they take up the spinal cord is called the spinothalamic tract. This is part of the anterolateral system, but the spinothalamic tract is the main important part there. So this is one tract within the spinal cord. They go up to the ventroposterior lateral nucleus of the thalamus, or the VPL, and they synapse onto neurons there. And then from there, those neurons in the thalamus project to the somatosensory cortex, the same parts of the cortex that we talked for touch and proprioception, so that we can perceive where in our body is pain being sensed. And you can see that this is being true to the map, right? So this light blue pathway for the lower body is that part of the thalamus is going to synapse onto the medial portion of the somatosensory cortex, where the lower limbs is located. And this neuron, the dark blue neuron that's on the upper body, is synapsing up here where, say, the arm is located. The arm or the hand, right? Which is going to be here on the more lateral portion of the primary somatosensory cortex.
So information from mechanosensation and nociception clearly takes different pathways. So this is a figure to sort of illustrate where in the spinal cord we might find information from these different pathways. And the essential pathways to convey somatosensory information from the body to the brain. Nociceptive information travels under a different pathway from mechanosensory input. That should be very clear in that the nociceptive afferents, these peripheral neurons, pain neurons, they synapse on these neurons in the dorsal horn. And then those neurons decussate at that level of the spinal cord and then go up the spinothalamic tract. also known as the anterolateral column. Mechanoreceptive afferents, on the other hand, actually stay on the same side of the spinal cord and then go up all the way to the dorsal column nuclei in the medulla. Thus, you have information coming from, say in this individual, the left side of this person's body, going up the left side of their spinal cord, and then pain information from the right side of their body going up the left side of their spinal cord. Thus, if there's damage to one side of the spinal cord, that will have a very unusual and a very odd influence over pain and touch and sensation. So if there's damage to just one side of the spinal cord, what's going to happen is that information from everything below that area, so let's say there's a cut right here in this part of the spinal cord just on one side, this individual will lose touch sensation on the left-hand side of their body and pain and temperature sensation on the right-hand side of their body. So they will have a loss of sensation, but on opposite sides of the body. Super weird. And that would be only if there's a transection on just half of the spinal cord, or these two pathways. If the transect was across the entire spinal cord, like most spinal cord injuries actually has, then information is going to be cut off on both sides, right? There are pathways on this side too, where nociceptive information on this side is carried up here, and then mechanoreceptive information is carried on this side here. So if you cut off all of it, then pain and touch will both be cut off on both sides, and you wouldn't have this weird split in the pain and touch information.
So just to re-emphasize this, and I feel like this figure actually conveys an awful lot of how to think about these pathways, and I also think is a great way to draw out and schematize the connections. So like this would be kind of an ideal way of reviewing how to draw diagrams like this. Little circles with a line and then a synapse drawn like that. Little triangle, sometimes just drawing it without the little line there, and instead it's just like a V shape. That's perfectly fine too. All right, but let's walk through this again. So the tactile proprioceptive information is conveyed through the dorsal column medial lemniscal pathway, right? So that's this, and it ends up information going through the dorsal root ganglia. There is some synapsing that does happen in the dorsal side, but the information, these are secondary afferents. The information is conveyed all the way up to the dorsal column nuclei in the medulla, and then you have the decussation event happening in the medulla, right? Pain sensation is conveyed through the spinothalamic tract, also known as the anterolateral system. And there is decussation that happens at the dermatome that is that part of the spinal cord is innervating. So across you'll have the afferent of this pain neuron synapsing onto a neuron in the dorsal horn, and that's the only place where it synapses. And then it goes, and then you have a separate neuron there that then that decussates and then carries information on the contralateral side through the spinothalamic tract, and then goes past the medulla and then connects directly to the thalamus. So there are secondary order neurons in both pathways, but one is up in the medulla for the mechanosensation part, and then for pain in the spinothalamic tract, it is in the spine and goes all the way to the thalamus, hence spinothalamic tract. Whereas in the medulla, it connects up at the dorsal column. So the dorsal column medial lemniscal pathway, you have information going up the dorsal column of the spinal cord, connecting to the dorsal column nuclei, and then those neurons go up the medial lemniscus, which is a pathway up to the thalamus. Hence dorsal column medial lemniscal pathway. So there are a few hints in there on how to memorize the anatomical locations, as well as memorize which pathways And again, I think this is an excellent way to schematize this kind of information. So this would be one great way of practicing and drawing out these pathways and understanding the difference between the two. Okay, so we have talked about sensory reception at this point, right? We've got these 4 basic steps. Sensory reception. We've talked for pain. We've got the the temperature-sensitive receptors. There are additional receptors as well that just sense certain kinds of stretching, but will convey pain information. Then we have the transduction of that information into opening or closing channels. In this case, say chemical or heat or shearing or acid will cause an influx of ion channels through those receptors. That's the sensory transduction part. Then we have transmission, which we just talked about, the spinothalamic tract. And then the last thing is perception. So
Pain perception is influenced by a whole host of factors, including other non-painful sensory information that enters the brain along with painful input. For instance, something scary that goes along with pain might modulate how you perceive that pain. Or let's say something funny is going on, that might actually tend to blunt the pain perception. So the perception of pain can be influenced by a lot of other factors than just, say, having a nail jabbed into your arm, right? There are a lot of other factors that can be taken into account that will modulate how pain is perceived. We're not going to talk about that cognitive modulation of pain perception in this presentation. It's just too complex for what we're talking about. Instead, what we're going to talk about how pain perception can be regulated in the basic circuits that we understand that regulate pain. So pain is an important thing. It's an important feature that all individuals need in order to protect their bodies and protect themselves from danger. But you can have too much pain too, and there are times when pain needs to be modulated and regulated. And we have 2 basic pathways in which pain can be modulated or regulated. One is referred to as ascending regulation, where there is regulation that happens with the input of mechanosensation and nociceptive information within the spinal cord itself as it's ascending up into the brain. So you can have modulation of the pain signal that eventually makes its way to the brain that is modulated here in the spinal cord. There's also descending regulation, where higher brain regions, such as in particular the periaqueductal gray area, the PAG, which is right here around the aqueduct, that's connected to several higher order brain areas as well as a number of brainstem nuclei that then regulate pain perception via the release of special neurochemicals called endogenous opioids that will regulate how pain is being sent up to the brain via this descending pathway that comes down from the brain down into the spinal cord. So it's descending this way, and the ascending pathway is going up this way.
ascending regulation of pain sensation is, like I said, it's in the previous slide, it's controlled in the spinal cord itself. And there's going to be some additional details to the mechanosensation side of things that I didn't discuss in any detail in the previous lecture. So there's going to be some new details and additional complexity that's happening in the spinal cord itself. So in the dorsal horn of the spinal cord, we have inputs of mechanoreceptor information as alpha or beta axons. We also have these C fibers that are coming in, and this is the nociceptive signal. So we got mechanosensation and nociceptive signals. The route that we've already talked about for mechanosensation is that it goes down into the spinal cord, dorsal horn, and then it goes up the ipsilateral dorsal column. The C fiber connects to neurons that are in the dorsal horn, and then those neurons decussate and then go up the spinothalamic tract. Those are the 2 pathways that we've talked about. There are some additional afferents that mechanoreceptor neurons make within the spinal cord, though. They will synapse onto this population of interneurons. So both fibers, mechanoreceptor fibers and nociceptive fibers, they will make these collateral synapses. This is what that's called when you have a little additional synapse that comes off of a fiber, but it continues on and connects someplace else in the nervous system. These collateral synapses, they synapse onto this population of inhibitory neurons that are in the dorsal horn. So just to give you some context, we are zooming in on this part here. This is the dorsal horn of the spinal cord. And we're looking at this individual inhibitory neuron here. This synapse from the mechanosensation drives, it's excitatory, and it drives this inhibitory neuron to be more excited. The nociceptive signal, however, inhibits it. So there's going to be some sort of balance that's being done here. These inhibitory neurons then synapse onto that nociceptive projection neuron, the second order neuron of the nociceptive system, that goes into the spinal cord, decussates, and then goes up the spinothalamic tract. This inhibitory neuron then can serve as a gate for preventing pain signals to get up to the brain. And the way that gate can occur is that if there's activity coming through the mechanosensation side of things, mechanoreceptors being activated, it will activate these inhibitory neurons. Those inhibitory neurons then will inhibit the second order nociceptive neuron and prevent pain signals, or at least reduce pain signals from getting up into the brain. So you may have experienced this yourself. You might have had an injury, fell down, hurt your leg, and you might have had an impulse to kind of rub that area where it was hurting. That impulse, in fact, is one way of having this ascending regulation of pain sensation. That when you are rubbing there, you are activating those mechanoreceptors at that spot, and that you are then activating this inhibitory gate. And so then this inhibitory neuron would be activated, and it would reduce the amount of pain getting up to the brain. Is one way. It's obviously not going to cure anything. It's not going to prevent infection, injury, that kind of thing. But it can be one immediate way of regulating pain, is by providing pressure or movement, touch onto those areas in order to prevent pain signals from getting up into the brain. The second way pain is regulated, pain sensation is regulated, is by a descending regulation of pain sensation. As I said, there's a complex set of networks where information comes down from the brain through a number of brain areas, and it makes its way through the periaqueductal gray matter, which is located up in the midbrain. So periaqueductal means around the aqueduct. The aqueduct is one part of the ventricular system, and so this is an area that surrounds the ventricular system in the midbrain. It's called the periaqueductal gray matter, or the PAG. This is a critical node in this system. As you can see, you got several inputs coming into the PAG, and then the PAG sending projections to a whole bunch of other additional areas that are in the midbrain and brainstem. These areas then project down to the dorsal horn of the spinal cord to then regulate pain at the spinal cord, preventing the inducing changes in pain perception. The details here, I don't expect you to have memorized. I don't expect you to know the 4 different downstream nuclei. If you're interested in pain, if you're interested in knowing more about pain, do study up on it, read about it. There's a lot of interesting information on the internet on this, and it's worth looking into. There's also a lab within the School of Neuroscience that studies this regulation of pain at the level of the spinal cord with the interaction of ascending and descending. It's Anne Gregis's lab, so she's a new faculty member here at the School of Neuroscience, and this is the major center of her research topic. So if you're interested in learning more, you can go to her website and look up some information, read some of her papers, and if you're lucky enough, take a class with her on pain. So a lot of great information on pain. But what I do want you to take away from this is that the periaqueductal gray matter is the key gateway of information coming down from the brain, going through the periaqueductal gray matter, and then down all the way, eventually making its way to the spinal cord. So it's a key node in the system. Okay. So just to reemphasize that we have these higher order brain areas, sending efferent projections down to the PAG, which sends its inputs down to brainstem nuclei, regulating information in the periphery. One of the key ways that the descending pathway regulates pain sensation is via endogenous opiates. So we have chemicals in our bodies that act on opiate receptors. These chemicals that act on opiate receptors are the endorphins, and they will act upon those opiate receptors and regulate pain information. We call these things opiate receptors because, like a lot of receptors in the brain, they are first recognized as having anything to do with the nervous system because of, say, drugs of abuse. When we're discovering how they actually work in the nervous system, the first thing we find out is that opiates bind to these receptors, therefore they're known as opiate receptors. And there are a bunch of different common opiates. Morphine, codeine, and heroin are some that are used in in a variety of contexts. Morphine and codeine are naturally occurring. They come from opium, and they are used in a medical context primarily. Often they can be abused, but they are used in order to regulate pain. So from a surgery, from an injury, a toothache, you might be prescribed morphine or certainly codeine to regulate that pain. And it would be acting then on a series of brain areas that have opiate receptors, including the periaqueductal gray, areas of the thalamus, and in the spinal cord. So it'd be acting on all of these areas. Of course, we also have heroin, Percocet, and fentanyl, which have, and I think, and even then, still to this day, occasionally you might find heroin being prescribed, I think. But certainly even fentanyl is prescribed, but has now become, unfortunately, a drug of abuse because of its potency, and in fact is one of the major causes for the opiate epidemic of people dying from overuse of opiates. Primarily they are overusing fentanyl and unfortunately dying because of just how potent it is. So again, I don't necessarily expect you to have all of this memorized, where the opiate receptors are located, except for the fact that the periaqueductal gray is one of these areas, as well as the dorsal horn of the spinal cord is an additional area that has opiate receptors that are located there that will allow for activation of these receptors to regulate pain. And it is neurotransmitters that are regulated in the descending system that So a little bit more about these opiate peptides. The way this works is that the brain produces these endogenous opioids. Some examples of these are the enkephalins, endorphins, and dynorphins. They are released from neurons. They get transported down neurons and then released from the axons onto brain areas that have opiate receptors. The periaqueductal gray also it synthesizes its own opiates, and as well as other brain areas. And virtually all of these areas of the brain that have this opioid regulation are areas that are involved in nociceptive information. It's not the only thing that they do, but it is one of the main, that includes a lot of the main pathways that is involved in processing and sensing and regulating nociceptive information. So the opioid peptides, they can modulate pain sensation by directly inhibiting the ascending pathways. So this is showing some of these direct projections onto dorsal horn of the projection neurons. So this would be the C fiber coming in here. This is on the inside of the other side of the dorsal horn. So this would be like the left side. We were previously looking at the right side. So these C fibers would be synapsing onto the second order neurons that would then decussate and then go up the spinal cord. You have all these projections from several of these brainstem areas like locus coeruleus, the raphe. Again, you don't need to have those areas memorized, but these areas will project down into this specific circuit and release endogenous opioids that will act upon these neurons to modulate pain sensation directly in the spinal cord. But they are descending down from the brain with the paraqueductal gray serving as a central node. I wanted to finish up this lecture talking about an interesting, although relatively rare, phenomenon around pain, and that is referred to as CIPA, or congenital insensitivity to pain with anhidrosis. This is a genetic disorder that is quite rare. There are families that carry the mutation amongst Bedouin Muslims, so it's like in North Africa and in the Arabian Peninsula, where you're more likely to find this genetic disorder with a particular mutation. That when in those individuals that have that mutation, they will have the inability to form pain and temperature peripheral neurons. Those neurons are just missing. They also, for many of these individuals, they can't even sweat. So these individuals, they have a mutation in a receptor that's really important for how these neurons get wired up and connected. It also apparently is involved with sweat as well. So for those individuals with that particular mutation, they cannot perceive pain or temperature because they are missing those neurons. It turns out that the sweating issue is, for those particular individuals, tends to be the most serious issue, and that hyperthermia is a major cause of injury or death, and in fact 20% of affected individuals die by the age of 3 from hyperthermia. So yeah, if you can't sweat, you can't regulate your body temperature as effectively as you should be able to, and since of course this is a mutation that seems to affect populations in the Middle East, it's kind of hot there. It's really not a great combination. But it is a very rare condition. These mutations do arise somewhat spontaneously as well, and there are individuals that do have other forms of congenital insensitivity to pain. They don't all necessarily have the anhidrosis part. So I included a 60 Minutes clip. This is 60 Minutes Australia, where they interview 2 girls in the United States who have this condition, where they cannot feel any pain. So I encourage you, if you're looking at the PowerPoint file, to click on this link. I will also include a link in the description on YouTube to this video. Really interesting video talking about these girls' experiences. And one major issue for people who cannot feel pain is that they have a very high preponderance of accidental self-mutilation. Pain is absolutely needed for, as the father says in this video, a warning system so that you know to stay away from certain things and that you can avoid touching a stove, poking yourself with a sharp stick or sharp object, hitting yourself over, running into something. Pain is the key to knowing that you are actively injuring yourself. It's also the key to telling you to not do those things in the future. It is the negative reinforcer to say, you know what, stoves are hot. I shouldn't touch stoves. And so you'll see with these kids in particular, but this is true for anyone with this condition, they suffer through all kinds of injuries throughout childhood, or especially early childhood. They tend to have major joint problems. This is true for these individuals with the anhidrosis, as well as these kids as well. And that's because our bodies can't always stay in the same position or be in a position that is cutting off, say, circulation or is stressing a joint for very long. We can do it for a little while, but our bodies will tell us, hey, you know what you're doing right now, you're actually kind of injuring yourself. You've been sitting in this position for too long. It's starting to hurt. Your body, you won't even think about it, but you will be experiencing that pain, and your body will tell you to shift and move because you are actually likely to do damage if you continue to stay in that position. These kids don't have that sensation. They don't have the ability to feel that sensation. So they have major joint problems. They tend to have very large, swollen joints because of this condition. And even these things will happen at night. So we have an ongoing pain sensation that occurs at night when you're tossing and turning. Some of that is because of maybe the way that you're laying is causing some pain, and your body is saying, you know what, I should probably move, and you don't even have to think about it. Your body's just going to just send a signal to say, toss a little bit so that you are not stressing that joint, or you're allowing more blood flow to this area. And so a lot of these people have a very short lifespan because of these repeated injuries and that make them so susceptible to infection and other things. So that's where we're going to leave it. Here are the key questions. I'm not going to read them out to you here, but we covered nociceptors and the trip channels. We went through the specific anatomy of the nociceptor neurons, those primary nociceptor neurons, and I want you to know how are they similar or different from mechanoreceptor neurons. You need to know about the major fibers that relay pain and temperature information and how they differ from mechanosensation, how they go up the spinal cord, and knowing the anatomy and connectivity of pain information from receptor neurons all the way up to the cortex, and how that's different from mechanoreceptors. Which part, where does decussation occur, that kind of thing. And then also, how does the brain regulate pain information, and what are the difference between ascending and descending pain regulation pathways? And then, of course, opioids, how do they regulate pain? What are some of the specific spots? And what are some of the endogenous opioids that do that? All right, until next time, we'll see ya.